swine vesicular disease and vesicular exanthema of swine. So for swine vesicular disease, the occurrence, so Europe is free. It is a sporadic infection due to imports. It was found in Italy and Portugal and was first detected but was suspected to be foot and mouth disease. It's a rare disease. The OIE has reported no cases between 2015 and 2020. For the etiology of the disease, the swine vesicular disease virus is a part of the enterovirus genus, which is a part of Picornaviridae. There is only one serotype, so the Coxsackie B5 virus started affecting pigs, and now this new virus cannot affect humans anymore. The swine vesicular disease virus is not as severe, but has the same clinical signs as foot and mouth disease. For the epidemiology, it's stenoxin. It only infects pigs. And the virus is spread in animals, the environment, and products, and it's shed for 4 months. Asymptomatic animals can shed the virus. For the pathogenesis of the disease, oral infection is the primary way of infection. Then the primary replication happens in the tonsils and the gut. During the primary replication, they're asymptomatic, and there's no diarrhea or seroconversion, and the virus can shed before clinical signs. It is shed in discharges that can get into the environment and into the food. Then a viremia can occur, and the secondary replication happens in the epithelial cells at the predilection sites of the mouth, feet, and teats. On the mouth, it's the mucosa of the oral nasal cavity, and it's on the skin of the feet and the teat. There are also central nervous system signs, and it can affect the fetus and it can cause abortion. For the clinical signs, it's frequently asymptomatic. The incubation period is 3 to 7 days. The virus can cause fever, anorexia, lethargy, and lameness. And vesicles develop on the mouth, the feet, around the coronary band, which causes the lameness in the feet. And there can also be vesicles on the teat. These vesicles rupture after 1 to 2 days, and it makes erosions. Vesicles are the main clinical signs. For the pathology, the animals do not die. There are no gross pathological lesions. For the diagnosis, it's a notifiable disease due to the similarity of foot and mouth disease. The clinical signs cannot be distinguished from foot and mouth disease, so lab diagnostics is necessary. The virus can be detected directly through real-time PCR. It's the easiest and fastest way, and it can be detected indirectly through ELISA and viral neutralization. There needs to be clinical signs for the antibody to be present, and absent antibodies are needed for a country to be declared free of the virus. For treatment and prevention, there is no treatment and there is no vaccination. For the control, it's a notifiable disease in some countries, such as Hungary. It's not OIE listed. So for controlling the virus, you need to close the herd and have movement restrictions. And to prevent the introduction of the virus, ban swill feeding or slop feeding and control pig products. And if there's a case of an introduction or an outbreak, you need to have stamping out. There's also the Seneca virus A, which also causes vesicular disease of swine in the USA and Asia. It affects piglets under 1 week old and they die. And in adults, they have vesicles on the legs. The next disease is vesicular exanthema of swine. It occurs in the Pacific coastal region of the United States. It's not in the EU. When it first occurred, it was seen as an atypical form of foot and mouth disease. So the etiology of the disease, the vesicular exanthema swine virus is a part of the Vesivirus genus, which is part of Caliciviridae. The virus has more than 30 serotypes, as it's an RNA virus, which means it has high mutation. For the epidemiology of the disease, it's endemic in sea lions. They're the natural host. It gets into pigs via ingestion of contaminated fish meal. The susceptible hosts are pig, dolphins, mink, monkey, humans. It's a zoonotic disease. The virus is shed by pigs in the nasal discharge, saliva, and it's transmitted within the herd. It can also be spread by raw pork products. And it's unable to survive in the environment. It's non-enveloped. It can survive in water for 2 weeks. The virus has a slow spread, as it's a subgenomic mRNA. And the differential diagnosis is foot and mouth disease, which is highly contagious and spreads rapidly. For the pathogenesis of the disease, the virus enters the body orally through contaminated seafood, and the primary replication happens in the pharynx, and then viremia occurs, and the secondary replication happens in the skin and the mucous membranes of the mouth, snout, and feet. The clinical signs are fever, anorexia, and lethargy, and there's also vesicles, similar to foot-mouth disease, on the mouth, the snout, and the feet, on the coronary band, where we can see lameness. The vesicles are benign and they heal, and there's also a high mortality in piglets. For the pathology, the animals don't die. For the diagnosis, the clinical signs can lead to suspicion, but they're very similar to foot-mouth disease, which is a notifiable disease. So lab diagnostics are needed. The quickest methods are PCR and ELISA. And for the differential diagnosis, the PCR can also detect swine vesicular disease and foot-and-mouth disease. For prevention, there is no vaccination. To prevent the disease, you need to heat treat fish meal before giving it to swine. And if there's an outbreak, restrict movements and try and eradicate the disease.